of neurons. These respond to chemical and physical stimuli depending on the type of receptor that's associated with them. They also conduct electrochemical impulses in terms of action potentials. They also release different chemical regulators such as neurotransmitters and neuropeptides, and also enable perception of our world in terms of our senses, also are important for learning and memory, and then control of muscle and glands. Most neurons are not able to divide, but they can repair themselves depending on the extent of damage. But there also are some neurons within the olfactory for smell that have been found to regenerate, as well as within the hippocampus, which is involved within learning and memory, and then also part of our limbic system for emotions as well. So also to remind ourselves is that neurons are divided into a central nervous system and then peripheral nervous system. So neurons from the brain and spinal cord make up the CNS, and then the cranial and spinal nerves make up the PNS. And so the two different types of cells for nervous tissue are neurons and then glial cells or neuroglia. And so those are what support our neurons. When we look at a structure of the neuron, so the cell body, which we have right here, is going to be what contains the nucleus, as well as with other organelles that we find, such as mitochondria, endoplasmic reticulum, and so on. And when we have clusters and groups, these are called nuclei within the central nervous system, and then they're called ganglia within the peripheral nervous system. Next, we have the dendrites, and so these are going to receive impulses and conduct a graded impulse that will travel toward the cell body. The axon then, what we have here, is what conducts action potentials away from the cell body and will terminate on the synaptic terminals or terminal boutons. So the axon can vary in length from a few millimeters, what we find within the brain, or to up to a meter within the peripheral nervous system that extend all the way down to our extremities, such as our toes. Axon is connected to the cell body by the axon hillox, where the axon hillox is where action potentials are generated at the initial segment of the axon. And so the action potential will then travel down the length of that axon. This neuron here, what we have in the pink, this is covered with myelin. And so myelin helps in jumping of action potentials, which we'll get to later on. And then these exposed areas between the myelin sheaths are termed as the node of Ranvier. And so here we have very high concentrations of both sodium and potassium channels so that we can generate quick action potentials. So within our neurons, we need an active process that moves organelles and proteins from the cell body to the axon terminals, and then also backwards as well towards the cell body. And what mechanisms we have are through microfilaments and microtubules and different types of motor proteins. And so there's two ways of transporting materials. The first way is anterograde transport. And so this is moving from the cell body toward those axon terminals. And the second type that we have is retrograde transport, where we go from the axon terminals back toward the cell body. And so we have different types of proteins also that are responsible for this movement. And so this is how we have transmission of different materials as well. But sometimes this is detrimental, where with retrograde transport, sometimes we can accumulate waste products or viruses that can affect them and damage the cell body. However, An example for anterograde transport is of mitochondria, where we need to have a lot of mitochondria when we have action potentials, so it helps with influencing of the neurotransmitter release. Three different categories or classifications of neurons, which are all based on the direction of impulses that are conducted. So first with sensory neurons, these conduct impulses from sensory receptors in the periphery to the central nervous system. Motor neurons conduct impulses from the central nervous system to target organs, such as muscles or glands. And then lastly, we have interneurons, which are found within the central nervous system only. And what they do is they integrate functions of the nervous system, and so they act as like an intermediate mediator as to what type of information will be transmitted into the central nervous system or into the peripheral nervous system. So when we look at this, what we have here is an example of a sensory neuron, and so it has receptors that will detect different stimuli, and so take that information from the peripheral nervous system and transmit it into the central nervous system. Okay? Somatic motor neurons will take information from the central nervous system and then travel it to our effectors such as skeletal muscle. So these are responsible for reflexes and voluntary control of our skeletal muscle. Autonomic motor neurons, what we have here at the bottom, help to innervate involuntary targets like smooth muscle, cardiac muscle, and our glands. And so we have two different types where we have our sympathetic neurons and then parasympathetic neurons, where sympathetic stimulate a fight or flight situation and response, and parasympathetic are important for normal functions such as rest and digest. So now moving on to glial cells, and so these are the non-conducting but supporting cells. Glial literally means neuron nourishing, and so that is the function for them. So we have two different categories of glial cells, two of which are found within the peripheral nervous system and four which are found within the central nervous system. So within the peripheral nervous system, we have Schwann cells. And so in this example, this would be Schwann cells here. And so these form myelin sheaths around peripheral axons. Satellite cells, which are found in the purple outline here, are important for supporting cell bodies within the ganglia of the peripheral nervous system. And so they help to increase the size of that cell body. Within the central nervous system, let's start out with oligodendrocytes. And so these ones, what we have here, within the central nervous system, form the myelin sheath around axons, so very similar in function in terms of Schwann cells for the peripheral nervous system. Microglia, micro meaning small, help to migrate around the central nervous tissue, and what they do is they phagocytize and degrade foreign and degenerated materials within our central nervous system. Astrocytes, what we have here in that green color, help to regulate the external environment of neurons, and then also helps to create the blood-brain barrier that we have, as well as a nerve blood-brain barrier amongst neurons themselves. And then lastly, ependymal cells help to line the ventricles and secrete cerebrospinal fluid to keep that environment nourished with plenty of nutrients and ions for our nervous tissue. So one unique feature that we need to talk about is myelination versus unmyelinated axons for neurons. So let's start with the peripheral nervous system, where all of the axons in the peripheral nervous system are surrounded by a sheath of Schwann cells, and that's called neurolemma, or the sheath of Schwann. These cells wrap around the axon to form the myelin sheath within the peripheral nervous system. So here we have an example of the myelin sheath around this axon in the PNS. So as a reminder, the gaps between the Schwann cells are called nodes of Ranvier, and so those are left open. Small axons, which are typically 2 micrometers in diameter or smaller, are usually unmyelinated. But even unmyelinated axons in the peripheral nervous system have some neurolemma, but they lack multiple wrappings around them to create that myelin sheath. So the purpose of having myelin is to conduct impulses very rapidly. So within the central nervous system, we have myelin sheath, but not neurolemma because we don't have Schwann cells, so it's a little bit different. So myelin helps to give tissues, or the axons, that white color, and that's what's the white matter because it's composed mostly of a lipid. The gray matter then is considered to be cell bodies and dendrites, which lack myelin sheaths. So the importance of the myelin, again, is to conduct rapid impulses of axons because the myelin creates such a high resistance at that point of the axon that the action potential, so here, let's draw just a little bit here, is actually going to jump at these nodes of Ranvier because it cannot penetrate then this myelin sheath. Okay, and so instead of having to travel the entire duration or entire length of that axon, it jumps, and so that conduction of that action potential is a lot faster. So typically, the fastest impulses that we have are myelinated axons and very large axons because that allows for a high concentration of those sodium and potassium channels to be within those nodes of Ranvier.